I was the last minute draft in, as I mentioned, and given I had much less time to prepare than all the other speakers, the quality is reflected in the talk, but I hope you stick with me. Now, I have a few different roles at the moment, but I've chosen the Sydney University PowerPoint template for this one because it's mostly work that I'm doing for my PhD at Sydney University. I'm at Royal Brisbane, Hurston Infectious Diseases Institute, UQCCR, the department, and a few other things as well. So I've got a few disclosures there, none from diagnostic companies though. So I've got a case study, and actually, Jessica, I've got a horrible worry now we've chosen the same case. So if do something like that, if I'm presenting the same one or something, like it's a pretty famous patient. Yeah. I'll stop if I'm cutting in on your turf. Let me know. I should have asked you first up. But so I want to present a case study possibly that presents some of the issues around what I was talking about here, and then introduce you all to a new concept in infectious disease diagnosis of metagenomics. Tell you what it is, what the applications are going to be, some of the approaches and challenges, and then have a bit of blue sky thinking about a wholesale change to the way we diagnose infections in transplant recipients in the future. And then I'm going to outline one of my PhD studies, which is actually an extension of the T2MR study that Neil already mentioned, but T2MR refers to a different diagnostic approach, and we're adding in metagenomics to that existing sort of ethics and governance process. So I think the general public has an idea that when you have an infection, or in fact any disease, that the doctors, like here in Star Trek, will be able to hold a little gadget next to the bed and within a couple of seconds will give you a diagnosis and may well include treatment as well. It's quite a high bar and one we haven't really achieved yet, but I think that maybe metagenomics is going to take us in a big way to furthering the ideas. And then, you know, at some point in your great-grandchildren's careers, we'll have caught up with public expectation. Now, I changed the names. This looking familiar? No, this is good. This is really good. Okay, so we're very, very carefully organised before the meeting here. So Jeff is a 42-year-old man. He had an allograft in 2014 and is the most immunocompromised patient in Queensland, possibly. He had relapses. He's had severe graft versus host disease with a lot of treatment, including ruxolitinib and oral corticosteroids. He has diabetes and is pretty unwell. He has had 11 admissions to Royal Brisbane between January 2023 and the present with various infectious diseases, notably an Elizabeth Kingia Anopheles bloodstream infection that went for 52 days. I was only on service for the first 31 days of that, so it's not totally my fault, but we had a lot of trouble getting that under control. He's also had multiple other bloodstream infections, the four most interesting ones I've listed there. He has an Aspergillus fumigatus invasive lung infection and actually went on our novel antifungal trial, the Olorifim trial, and continues on that. And he's also had at least 4 different respiratory viral infections with influenza B, parainfluenza 1, rhinovirus, and SARS-CoV-2. He also has repeatedly isolated some other organisms on his sputum, including a Scedosporium apiospermum, which we keep on getting in upper respiratory tract samples, but not on bronch samples. Lots and lots of Candida, a Penicillium or Talaromyces, and of course, CMV DNA positivity. So why do we need better diagnosis in haematology oncology settings? This is Louis Pasteur, who I think the joke's been used a bit too much now about how he could come into a lab now and be totally at home using modern day techniques. Like it's not really true. The computers would really throw him, I reckon, and stuff. And all the people on TikTok and stuff, and the women would be very confusing, I think, as well. But as you know, we still really rely on culture as a way of diagnosing infectious diseases. And whilst it's an excellent test and has a lot of advantages in terms of phenotypic characterization, it can be quite slow and the sensitivity can be poor. So we sort of have moved into the polymerase chain reaction world, particularly for RNA viruses and I guess viruses in general. And that's really been around for most of your careers, but not all of ours. Naomi, looking for you and the older members of the audience here. And then about 20 years ago, we started taking steps into genomics, first in the human world and then in the pathogen world, 16S RNA probably being the most successful of those. But the new kid on the block, the very exciting technology, it's been around for about 12, 15 years roughly now. I think the seminal papers from UCSF for about 12 years ago is the idea of metagenomics. And the broad concept here is instead of trying to pick an organism, make a diagnosis based on clinical suspicion or whatever, you just sequence everything, just find everything in the sample and sequence the a hell out of it and just see what's there. And it's super fun and interesting, and it amazingly works really well. So metagenomics is a step away from the idea of writing a pathogen on the request form. So you don't have to write SARS-CoV-2 on the request form. You don't, it's sort of a pathogen agnostic, non-bias approach to diagnosis. Hypothesis free is one I really like, although it makes us sound a bit dumb, like we don't have any idea what's going on. It has some strengths. Now, I could argue that I guess blood cultures are also an example of a syndromic test for bloodstream infection and sepsis, but this is taking it to a new level. Another huge advantage is it can be used to identify unknown, divergent, novel, emerging pathogens. And so SARS-CoV-2 was diagnosed by clinical metagenomics on a sample from a patient in Wuhan Hospital very easily within a matter of a couple of days after getting the sample. What metagenomics reflects is that, in fact, kind of concepts of infection are all a bit wrong, where we just, I guess, have an image that you find an organism in a site and it shouldn't be there because it's in high amounts, and that makes it the pathogen. What we know, in fact, is that there's a community of infectious diseases, or an infectome, in all of us all the time. It contains a mix of viruses, bacteria, fungi, archaea, whatever they are, and lots of host reads as well. And a lot of the time, it's very difficult. We find we're not very good at telling what the difference is between colonization and infection, infectious diseases. And I think the main reason for that is it's an arbitrary concept that doesn't actually really exist. There's generally an ecological approach to the idea of infection now. Metagenomics can tell if an organism is present or absent, and I guess that's one of the major features of my current study, is just to test the sensitivity and specificity. Importantly, it can give a quantitative result in reads per million, and that's a big advance over some of the other technologies we have, and that quantification is probably more accurate and reliable than most of the others we have in microbiology now. Along with the presence or absence of an organism, you can do things like characterize antibiotic resistance genes, virulence genes. Increasingly, with really, really high intensity sequencing, you can also start looking at what genes are being expressed by looking at the transcriptome through mRNA transcriptomics. You can do that for the organism, and I think very excitingly, you can also do that for the host. Because it's a profile of the entire organism, you also automatically get things like typing, which then allows you to explore hypotheses around transmission, disease linkage, outbreaks, reinfection, recrudescence. The current literature is pretty well summarized here. There are some studies really where a single group in a single hospital have got a sequencer and just gone to their ward and sequenced a whole lot of people. It's all really been primarily DNA sequencing, and it's been based on blood culture, comparing to an arbitrary standard of just what the results in the blood culture were, or potentially having a clinical adjudication panel that's attached significance to the result. That's actually really hard to do. And frankly, I think all of these studies that I've got here, there's six there, and there's probably about 6 or 7 more, they're all dodgy. Unfortunately, there's no really good systematic approach to it. I was starting to look into doing a meta-analysis of this, but I was just going to come up with one of those kind of wishy-washy conclusions about there not being enough data. So I thought I'd focus on that instead. All right, so in our clinical practice, metagenomics is currently used when we have perplexing cases, defying diagnosis by conventional means. Encephalitis is a really good example of that. Already, San-Marie Schlebusch and Amy and co can do a metagenomics panel at Forensic and Scientific Services for about $700 when you have a confusing case, and you just have to fill out an SAS form. It only includes DNA sequencing at this point, but it's already proving quite useful, I think. I guess the emerging role is syndromic testing. In this context, febrile neutropenia is the syndrome. And of course, we don't look after, we've never been merchants of looking after infections. We're always diagnosing a syndrome. And I think that's really important when you ask questions about the sensitivity of metagenomics. The sensitivity of metagenomics to pick up a particular organism is one thing. The potential of standard diagnosis to diagnose a syndrome is very different. And that's the actual clinical question. There is no other question of relevance. My big blue sky rocket, Elon Musk X to the moon type concept is, I think there's the beginnings of a glimpse that this technology could replace conventional microbiology entirely. I get really riled up when I say that, particularly the microbiologists. I'm not one, I'm a clinician. But that's why I do it, because it's fun. There are the glimpses of that beginning. I think that the consensus in the ECMID session is we're a long, long way from that, and there's a whole lot of hard work. But in terms of the cost of sequencing coming down so dramatically now, and the very, very wide availability of bioinformatic pipelines, it's all becoming a bit more kind of real than it maybe was 2 or 3 years ago. All right, so what is actually metagenomics? There are three sort of different domains, I guess. The first idea is whether you do an enrichment approach where you have some way of having some hypotheses about the organisms that might be present, then use a PCR or some other amplification step to try and amplify the DNA or RNA from those organisms and then sequence those, which obviously has much better sensitivity, versus this idea of shotgun metagenomics. That's where you just do everything all the time. The obviously big upshot with shotgun is there's no hypothesis required. You can be kind of just a bit vague like me these days and just kind of get everything. You don't have to overthink it. DNA metagenomics is by far and away the dominant. In fact, there are no studies that I'm aware of looking at RNA sequencing in hematology oncology populations. That's because that's where largely the technology has been. But I think actually RNA sequencing has a lot of benefits. And then I don't know if you've ever done a long hike in Tassie or New Zealand and everyone sits around discussing whether trangiers or gas stoves are better. In the genomics world, that's really people sitting around discussing whether Illumina or Nanopore are better. And there's never going to be an answer to that. And everyone has incredibly passionate beliefs depending on what they just invested in their lab last week. So there's a trade-off between sensitivity in general, the amplification tests using some sort of, often a PCR to amplify targets in your sample, improve sensitivity versus universality, universal application. Although that sensitivity issue is relevant for the organisms that are there, sometimes our targets don't actually match with the pathogens very well. I've got, from a different study I've got some really good examples of when you use very, very heavily multiplex PCRs, the quality of the PCRs really wears off with the number of targets. If we're looking for outbreaks, emerging pathogens, novel disease, or perplexing cases, really you want to know about off-target organisms. And I'm sure you've all got a spectacular diagnosis made by metagenomics that wasn't immediately apparent by your standard approach. I have to say, when I look at the current enrichment panels, they've clearly been done by the lab people because they're not really all that informed clinically. You'll get, these brilliant panels for respiratory infections that only include viruses and not mycoplasma, for example, or you might get these wonderful stool panels, which are just not of any clinical relevance to us. The downside, of course, with amplification-based approaches is they're not hypothesis-free. You do have a panel of organisms that you're looking for. What about DNA sequencing versus RNA sequencing? Well, DNA sequencing has been around for longer. It's much more established. DNA as a nucleic acid is much more stable and much easier to work with. It doesn't degrade as quickly, so you can actually dig up bodies from 5,000 years ago and sequence their hepatitis B virus DNA and work out that they had a particular type of hepatitis B, whereas RNA is very unstable, one of the big downsides of it. RNA sequencing is the new kid on the block, but I do think that more and more groups are switching over for some really good reasons. RNA is very unstable. Every cell, every human, animal, plant cell has RNases that immediately start breaking it down. There's a lovely paper from the from these like forensic medicine CSI type literature saying that RNA ex vivo has a half-life of 4.5 minutes, so it's pretty hard to handle. Obviously, RNA viruses are really important, and the only way of picking up RNA viruses really is to do RNA sequencing. It's actually fantastic for detecting bacteria and fungi as well. Of course, bacteria, fungi, eukaryotes, us, we're all producing RNA all the time in very high abundance. And so it's actually a really good way of detecting it. Potentially, it might even be better than detecting DNA because when you get mRNA present, it means that an organism is actively transcribing and producing proteins, i.e. maybe it's actually doing something, causing an infection rather than just sitting there. And we've got the first glimpses of that in our own data now. The other thing you can do with RNA, of course, is that we all produce RNA when we get an infection as well. I won't go into that in too much detail, except that there is a group in UCSF in California that's really nailing this and is beginning to use machine learning algorithms to look at the host transcriptome and predicting with 98% sensitivity and specificity whether a patient has a bacterial, viral, fungal infection, whether they have an autoimmune disease. And they've made some funky diagnoses, things like sarcoidosis and Voigt-Harada-Kanagi syndrome based on the transcriptome. There's also a group in Korea that's now started successfully diagnosing lung cancers on BAL specimens using the transcriptome as well. I guess that one of the big barriers often put up for metagenomics is bioinformatics. It previously was thought to be scary and a massive barrier because of the complexity involved, the expertise, and the massive expense of high-performance supercomputers. However, I have to say that's really changing very rapidly now. What we have now is a lot of pipelines. There are commercial pipelines, and you can now literally, for a cost, send your FASTQ files to a company and they'll give you a report 24 hours later with everything you could possibly need to know. There are lots of cloud computing options available as well. In the picture here, I've got a picture of the Ford cluster at UQCCR, which is the HPC that we have on site. We also have access to a few others. That's the Pawsey supercomputer in Western Australia that is not only a fantastic machine, but also very, very environmentally friendly and has low carbon emissions. But increasingly, lots of other groups are looking to use cloud-based services like Amazon Web Services, which have the huge benefit that you only have to pay for the time you use. These supercomputers have to be switched on all the time and you pay for them whether you're actually sequencing or not. I just put a little picture of my M3 Mac chip down there because actually it can now do a genome assembly pretty amazingly. As long as I don't want to do anything else for the day, it can do some of this work itself now, as long as you've got enough RAM. And for the Oxford Nanopore side of things, there is now on-device there's on-device base calling and bioinformatics through Min now, which you can see here, just calling the bases in real time. So the other big step there is that artificial intelligence and natural language processing is making a huge difference. And really, I would use AI, ChatGPT all the time, every day in my PhD to help with coding now. And it's making it much, much easier. And it's just getting better and better over time to a point where you could really imagine within a couple of years, you'll be able to have a natural conversation with a machine and it will give you the data that you want. I mentioned on-device bioinformatics already, but the big benefit of those is real-time results, including some diagnoses now of say SARS-CoV-2 infection within 40 minutes of it going on the device. Some of the challenges with metagenomics relate potentially to the sensitivity. So remember, for shotgun metagenomics, there's no amplification step. It's just what's there, and the RNA might be quite badly degraded. However, I think with improvements in sequencing, one of the approaches that we have adopted is to just do a whole lot of sequencing. So we access the NovaSeq X Plus, which has ridiculously massive amounts. So you get about 8 gigabytes of data per lane. Now we're getting up to, in the case I'm just about to show you, we're getting up to over 170 million reads per sample now. So even organisms in very, very low read number will still pop up. The specificity and the interpretation of organisms in low read count. So when you do these panels, you will get a list of about 200 organisms on the list, has been really held up as a big problem. There were some really nice mathematical solutions to this problem presented at ECMID this year, ways that you can mathematically interrogate the data. And I guess it's a question of the maths now really catching up with the data to show techniques for improving the specificity of your results, like getting rid of the chaff whilst really minimally affecting the sensitivity. There is this idea of index hopping that when you multiplex samples with a very, very dominant sample, you can get a bit of movement of a sample within the run into other libraries, which can confuse things. And the databases are still, at the end of the day, pretty low quality because they haven't very, very well curated, even the official ones. The cost was an issue. I really see that as becoming less and less of an issue. So I don't think the BioFire film array respiratory panel is around the order of around $400 now. And we think we can get our metagenomic panel out for about the same now, so around $400 or $500 for a panel. So it's starting really to compete with multiplex PCR. Remembering a gene expert 4-plex costs about $85, and for a smaller read number, for just a quick and dirty, we think we're pretty close to getting around the $100 mark for something very quick in the centre. A lot of that expense actually relates to multiplexing and throughput because these new instruments are so massive that if you can fill them up and use them, the actual cost per sample per run is actually very low. So just a bit of blue sky thinking quickly before I wrap up. The host transcriptome is very exciting. I've sort of been through that, but I've just been quickly finalising an ethics submission for that. We'll start looking at host transcriptome as well. We have been carefully bioinformatically cleaving away all the host RNA, but actually there's a wealth of information in there. There's an option now, and some groups are already doing this, looking at pre-infection metagenomics. So there's a study in hematology oncology doing metagenomic sampling prior to febrile neutropenia with a very good ability to then predict what the bacteremia is and giving empirical antibiotics even before, you know, it's intelligently based on what was there a couple of days before the fever, up to 7 days before the onset of fever, with better prediction than just doing the empirical treatment. I sort of mentioned the work around machine learning already, so I'll skip through that. Okay, so we took a respiratory sample from Jeff, our patient, back in January. He'd been admitted with a nasty, what looked like a viral infection, and we found the parainfluenza 1 virus that was also present on his PCR, his extended spectrum PCR. And you'll see here, so that respiratory virus up the top there is the parainfluenza virus in very, very high read count with a very good ID match. The rest of the results here are pretty fun and interesting, and I've just sort of cut the top 10 here, really, but there are lots more to go through. All these Partidiviridae are fascinating. They're all Aspergillus phages, and we found a huge abundance of viruses infecting his Aspergillus, which we also found here. So these are Aspergillus fumigatus. Amazingly, on the RNA sequencing, there were thousands times more phage RNA copies than Aspergillus copies, but we managed to find that very easily. It's a very clear, obvious diagnosis. Moving down, he'd only had a Stenotrophomonas bloodstream infection a couple of weeks before and was still recovering from that, and we found Stenotrophomonas DNA as an RNA in quite a lot of abundance. We also found his Enterococcus and the Elizabeth King ear was still there floating around in his lungs, even though we haven't actually seen that clinically for about 10 months. The Klebsiella and all of the others were there as well, suggesting that, in fact, what this man has is very heavy pulmonary colonization with a whole raft of different organisms, and he's constantly resetting his bloodstream because he's never able to clear it. We have actually just put his stool on the sequencer this week as well, so we'll be looking to see if there's a reservoir there, and are contemplating offering him a stool transplant, fecal microbiome transplant, if we find large abundance of the Elizabeth King ear and other organisms. I won't show the corona plots for the sake of time. So as Neely mentioned, we've got the T2MR study at Rural Brisbane, but we've now expanded that out to include metagenomics. And we're going to do a multimodal novel rapid diagnosis of the cause of febrile neutropenia in haematology patients. I'll be specifically looking at respiratory samples and specifically using RNA sequencing, and we'll be running those on both the Illumina long read system and also on the new PromethION P2 Solo Oxford Nanopore, long read system and the Illumina short read system. So it's a comparison, really assessing things like sensitivity, specificity, turnaround time, predictive accuracy. And we're setting up to get a validated test. We've got funding for a clinical fellow in Pathology Queensland to work on the validation and NADA accreditation for an in-house test that we can then subject to a clinical trial in the future. I just mentioned quickly that we've put a lot of effort into this, and we realised that really you do need to handle RNA carefully. So we now collect our samples into RNA later. Or in the case of bronchoalveolar lavage fluid, our respiratory service collect those into dry ice for us, and so they remain very, very cold, and we preserve the quality of the RNA as best as possible. Skip all through that. So thank you. That's a quick glimpse of what we're doing. When I hopefully give this talk in 2 years' time, we'll have a few hundred patients with some clinical data, but certainly the first about dozen patients who are coming through are looking really, really promising in terms of the sensitivity being really just as good as conventional diagnosis. I'd like to say thank you to the patient and his family. He's had a very, very rough time, but has been really generous in letting us access samples and doing some public speaking for us, and the team that look after me at University of Sydney, UQCCR, Heidi, Haematology at Rural Brisbane, and all of the ID consultants and registrars at Rural Brisbane. So thank you very much.